Over the past 15 years, CRISPR has revolutionized the biotechnology world. In this video, we're going to explore the incredible potential of CRISPR for gene editing and how it works. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are special DNA sequence repeats found in bacterial genomes. This region is associated with a protein called CRISPR-associated enzyme, or Cas, which can cut DNA. In bacteria, the CRISPR system helps the cells defend against invading viruses by cutting the virus's genome. Scientists have harnessed CRISPR's abilities for use in the laboratory, turning it into a powerful and precise gene editing tool. With CRISPR, we can precisely cut and modify DNA sequences using a few key components. To use CRISPR for gene editing, we need three main components: a guide RNA to target specific genes, the Cas enzyme for cutting DNA, and DNA from the organism we want to modify. Let's start with the guide RNA. The guide RNA is like a GPS for the Cas enzyme. It leads Cas to the exact spot in the DNA that needs to be cut. In bacteria, the guide RNA has two parts: tracker RNA and CRISPR RNA. The tracker RNA, or trans-activating CRISPR RNA, is a long piece of RNA that folds into a stem loop. Once bound, the loop activates the Cas enzyme so it can cut DNA. Importantly, the tracker RNA stays the same no matter which DNA sequence you're targeting. The CRISPR RNA is what makes CRISPR gene editing so specific. It's a small RNA molecule, typically 17 to 20 nucleotides long, that complements the target DNA sequence. Each target sequence has a tiny sequence called the protospacer adjacent motif, or PAM, immediately downstream of the target sequence. This determines which Cas enzyme can cut the DNA. When picking a target in the genomic DNA, scientists make sure there's a PAM site in the right place and that the CRISPR RNA matches the DNA. It's important to check both the 5' and 3' strands of DNA during target selection, because the enzyme can cleave either strand. In the lab, scientists often combine the CRISPR RNA and tracker RNA into one piece called a single guide RNA, or sgRNA. This simplifies the process by eliminating the need for multiple RNA components. The next component is the CRISPR-associated enzyme, or Cas. The Cas enzyme has different domains that help it target and cut the DNA. One domain recognizes and binds to the target DNA, another recognizes the PAM sequence, and the last domain cuts the DNA. Finally, we need to prepare cells that contain the DNA we are modifying. This involves transforming the cells with a DNA construct containing the CRISPR RNA and the Cas enzyme. Once inside the cell, the sgRNA and the Cas enzyme are expressed. The complex looks for a match between the RNA and DNA. When it finds a match, the Cas enzyme cuts the DNA, triggering the cell's repair mechanisms. There are two main DNA repair pathways used to fix the cut: non-homologous end joining, NHEJ, or homology-directed repair, HDR. The most common ways cells repair cut DNA is through non-homologous end joining. This process quickly joins the cut ends of DNA, but can be error-prone, sometimes causing mutations like small insertions, deletions, and frame shifts. This can lead to loss-of-function mutations, making NHEJ ideal for gene knockouts. Homology-directed repair, or HDR, uses a matching DNA template to accurately repair double-strand breaks. In the lab, HDR lets researchers insert large DNA sequences. They design a template with the desired sequence surrounded by matching segments near the cut site. When present in the cell with guide RNA and Cas enzyme, the template is used to repair the break and insert the new sequence. While CRISPR is powerful, it has risks, like mistakes in DNA repair or accidentally editing the wrong DNA. Ongoing research is important to understand and minimize these risks. Although it evolved as a defense mechanism against infection in bacteria, CRISPR has become an indispensable tool in biotechnology. It holds great promise for improving human health, food security, and environmental sustainability. As we continue to explore CRISPR, it is important to use it responsibly for the benefit of science and humanity. Thanks for watching. We pride ourselves on providing the highest quality customer service for our teachers. Call, email, or send us a message on social media.